See, students, in MSc third semester, you know, in the evolution portion, there is one major aspect related to Hardy-Weinberg equilibrium. And then in that chapter itself, it is mentioned that under what conditions the Hardy-Weinberg equilibrium can be maintained. So those conditions are actually uh, the mutation, selection, genetic drift, and migration. Means these four conditions must not operate in the population. Only in such condition, only then the population will be in Hardy-Weinberg equilibrium. So the major forces of evolution are actually these four. That is uh, mutation, selection, gene flow, which is also referred as migration, and genetic drift, which is sometimes referred as random genetic drift. So these are the forces of evolution, major forces of evolution. That is a population, sexually reproducing population definitely experiences these factors, and that is why every population of a species will be the subject of evolution. That is, it will definitely experience evolutionary changes. So let us go through these individual factors one by one, and then you can have the idea regarding change in gene frequency, because when we talk about evolutionary change in a population, we mainly consider change in the gene frequency, okay, which actually affects the Hardy-Weinberg equilibrium. So the first, major force of evolution is mutation. We all know that mutation means sudden genetic change in the uh, DNA molecule, and such change will be in the form of point mutation or nucleotide changes, because mutation uh, that can be categorized as point mutation or chromosomal aberrations. But here I'm mainly talking about point mutations. Sometimes people also consider mutation with respect to chromosomal changes, which could be structural changes and numerical changes. But when I refer mutation here in this respect, I am mainly concerned with nucleotide changes, and it is a change in the genetic material, that is DNA. We know that all organisms, they have their genetic material in the form of DNA, and that is subject to change. It definitely experiences mutational change. There are several forces. Such forces could be artificial conditions, or such forces could be induced forces. Even spontaneously mutations may occur in the genetic material. Spontaneously means at the time of DNA replication, the nucleotide changes may occur because of some error, although there exists a very good kind of proofreading mechanism. But even then, some sort of changes is likely to occur in the during replication process. And a number of other ways are also there through which spontaneous mutations are created, particularly in the germ cells, and then those changes may get inherited to the further generations. So they are such changes which occur in genetic material are actually heritable. They can be transferred from one generation to the other. It is unavoidable process, means this has to definitely occur. Mutation will be definitely occurring in the population. It cannot be stopped. It has to happen. A single nucleotide or several nucleotide changes may cause new allele formation. So this is an important point to remember, that mutation actually create new alleles uh, in the genetic material. One can say that if there is an allele, suppose capital A, then it can change into another allele like A1 or A2 or A3 only because of mutation. So it is mutation that creates new alleles, and such alleles may accidentally be favored and may come in the population even if they are not being favored, means they may get retained in the population. So mutation could be beneficial, that could be deleterious. So mutation alone creates new alleles in a gene pool. This is the point one should remember. Then it occurs randomly. Means you cannot say that this specific segment of DNA is more susceptible to a mutational change. No, definitely if it occurs in the well conserved regions of the DNA, then it will not be retained because such mutations may be harmful. So it occurs randomly, and there are areas where the mutation rate could be seen high because such mutations do not actually affect the fitness of the individuals, or they may not be lethal like in repeated DNA sequences. Mutations may be frequently occurring, and that is the reason that we have a large amount of repeated sequences in the form of microsatellites or mini satellites. So, but the conserved areas in the DNA which code for important proteins, they uh, do not actually accumulate mutations, because if they go on accumulating, then that will definitely affect the life of the individual. So, although such mutations occur randomly without regard for any possible benefit or disadvantage to the organism. We are quite aware with several terms related to this mutation, like silent mutation, means mutation may occur in the genetic material, but that may not influence the change in the amino acid. Okay, because if you see the codon, generally the third letter change in most of the cases does not affect the amino acid change. So silent mutations are those mutations which do not actually result a change in the amino acid sequence. So you can say it is a kind of synonymous mutation. Then the missense mutation, that is change in the nucleotide results into change in the amino acid sequence in the polypeptide chain. Then there could be nonsense mutation. If mutation occurs in the DNA, then as such, because of that, there will be a different kind of codon system in the messenger RNA, and that will result into termination of protein synthesis. So nonsense mutation may occur, means because of such mutation, the codons may come over there as UAA or UGA or UAG. So these are terminator codons, and because of such mutations, the protein synthesis will not be getting complete. So then frame shift mutation is there, means due to deletion or duplication of certain nucleotides in the DNA, there may be shift in the entire codon system. So these are the terms which I hope that you people might have studied during your genetics class. So there is no need to elaborate all these things. Only thing is that we have to consider that mutation uh, that cause mainly a change in the DNA molecule, and such change may result in the formation of new, alleles, or a particular allele which is quite prevalent in the population may get changed into another form. Now, whether we can see the mutation rate, answer is yes. Means several genes and the rate of mutation in those genes have been recorded. And in certain books of genetics, if you study this specific chapter mutation, then you may get a table where the genes human beings present in human beings or in other organisms, and their rate of mutation may be given. But I'm going to tell you that how such things could be known. See, if there is a mutation which is of recessive in nature, then it would really be difficult to find out its occurrence in the population, with what rate it occurs, because it is recessive in nature, until and unless you go on sequencing the DNA molecule from the large number of individuals in the population, it will not be possible. But this analysis can be performed in such cases where a gene is or an allele is dominant in nature. So mutation rate may be evaluated for certain traits, particularly the dominant traits. Those traits which are dominant in nature, their mutation rate in the population can be deduced. For example, if two births occur out of 1 lakh individuals in a population, see, 1 lakh individuals form a population, and in them two individuals are born with a dominant trait. Suppose two children get birth and they show a dominant trait. Definitely, if they are showing dominant trait, it means they are carrying the allele which is dominant one. So you can say two dominant alleles are present out of 2 lakh alleles, because 1000 individuals you have observed, it means 2 lakh, so I'm sorry, 1 lakh individuals you have observed in the population, or out of 1 lakh, it means 2 lakh, uh, alleles will be there. So out of 2 lakh, two individuals means two dominant alleles are being observed. So rate of mutation, here this should be a symbol mu, will be equal to, this rate of mutation is indicated by its symbol mu. That will be 2 out of 2 lakh. So it will be 1 out of 1 lakh individuals. You can say that the rate of mutation is 1 into 10 to the power minus 5, because 1, 2, 3, 4, 5, 0 are here. So the rate of mutation will be 1 into 10 to the power minus 5 per gamete per population. And this is the way, means for dominant alleles or dominant traits, this kind of analysis is possible. And in our own population, human population, achondroplasia, which is a dominant form of dwarfism, means dwarf people, they suffer from achondroplasia. So it is a dominant trait. And generally, those individuals who are dwarf, they are heterozygous, and they carry one allele that is dominant one, and that results into this kind of effect, that is they are dwarf individuals. So homozygous dwarfism or homozygous uh, individuals will not be surviving, because such conditions are lethal. So its rate has been observed in the population, and that has come 1.4 into 10 to the power minus 5, exactly following this population analysis or population data. This has been observed. So you should know that for certain traits, particularly the dominant traits, this kind of analysis can be performed, and one can know the rate of mutation. And since you people have studied this aspect in much detail, so I think that this much information is enough. Then the other factor which influences gene frequency in the population, or which affects Hardy-Weinberg equilibrium, is selection. And Darwin used the term natural selection, but here you can say selection as such will be better to consider, because this includes even artificial selection, that is the selection which is performed by human beings. So artificial selection, we people select economically important kind of plants, and even domesticable animals, uh, they are actually being selected by human beings. So selection as such, whether it is natural selection or artificial selection, that will definitely influence the gene frequency in the population. So what is selection? What is natural selection? Differential, survival and reproduction of different phenotypes or genotypes is known as selection or natural selection, you can say. That is, if you have, suppose, five different types of individuals, it means you have five different phenotypes. Suppose five different types of individuals means they could be different with respect to their phenotypes or genotypes. Now, it's not necessary that all those individuals will reach to their reproductive age. So those individuals who are able to reach to their reproductive age and produce progeny, they are considered to be selected. So this selection that actually that could be defined as differential survival and reproduction of different phenotypes is selection. Now, you people have already studied this aspect in case of industrial melanism, that is natural selection. We start studying by taking the example of peppered moth, that is Biston betullaria. So industrial melanism is one of the well-known case of natural selection we have already studied in case of peppered moth. Antibiotics resistance in bacteria, that is one aspect you might have studied. Then elimination of mutant type flies from the population, this can be another example. And since this particular case has been put in your syllabus, particularly in the uh, practical syllabus, I would like to explain this example, which is very nice one, and you will really be able to understand it. So I'm going to take up the case of natural selection by considering the case of mutant, Drosophila flies, which are cultured along with wild type flies, and we can see what kind of result we obtain in if we perform such kind of experiments. So let me explain this experiment of natural selection. First, you try to understand that what we do. We actually take up flies like red-eyed female flies. We collect virgin female flies from the wild type stocks. Then we collect virgin female flies from those stocks which are having white-eyed female flies. White eye is a trait that is situated on, or the gene determining this trait is situated on X chromosome. White-eyed females, they are those females which will have recessive allele, that is W allele, on their X chromosomes. So if a female which is having two X chromosome possesses W recessive allele on both chromosome on a specific locus, then those females will be white-eyed. So we collect, white-eyed females, then red-eyed males, and also white-eyed males. So let us understand that how the experiment is initiated. Suppose you have food bottles. Suppose you have just two or three food bottles, and in each food bottle you have introduced these 20 flies, that is 5 red-eyed females plus 5 white-eyed females plus 5 red-eyed males and 5 white-eyed males. So 20 flies you have introduced in the food bottle, in a single food bottle. If you have 3 food bottles, then in each food bottle you will have to introduce 20 flies in this number. Now suppose uh, in the next generation, means after 15 days, what you will do, you will first introduce these flies. So their genetic constitution. Those flies which are red-eyed females, they will have two X chromosomes, all those females, and on both the X chromosome, uh, normal allele, you can say W plus alleles are situated. Those females which are mutant, white-eyed, they will be homozygous. It means they will have two X chromosome, and on both X chromosome, WW allele will be situated. Those males which are red-eyed, they will have single X chromosome, other chromosome will be Y chromosome, and on the X chromosome, single X chromosome, the W plus allele will be situated. And those males which are white-eyed, they will have one X chromosome, other Y chromosome, and on this X chromosome, the W allele will be situated. So genetic, makeup of these four different kinds of flies you are knowing. Now, then what will happen after, means when you have introduced them in the food bottles, you will be waiting for 5 days, means for 5 days you will leave them to remain in the food bottles. After that you will remove them and you will get their next generation progeny. Suppose in the next generation you get flies of these four different phenotypes in this number, that is 455, 363, 460 and 390. Likewise, all these flies you have counted. See, this is the point where you have to take care. You will be counting these flies, and after counting them, you will have to transfer these flies in the next fresh food bottle. You will not, sort out any fly. You will all, means all these flies, all these four different kinds of flies which have been observed in F1, they will be as such introduced into food bottles, maybe 3, 4, 5 food bottles because they are more in number. So you are allowing them as such. You are not sorting out, you are not, minimizing the number of any type. Whatever number you get, you simply put them in the fresh food bottle and then try to get the next generation flies. In the next generation you have got again flies in this number. So this you will go on doing for, means up to that generation. When you find that the white-eyed flies or one specific kind of flies are not available. Although I have shown data only up to fifth generation, but such experiments have been continued for nearly 25 generations. And after 25 generations, the white-eyed, females and white-eyed males have not been obtained at all in the culture bottles. So what we observe that in further generations, the white-eyed females and the number of white-eyed males go on decreasing. This is the trend we observe. We observe that red-eyed females or red-eyed males, their number will not be affected to a to any level. There will be very drastic change in the number of mutant types, that is white-eyed females and white-eyed males. So here one more thing I would like to tell you, that after this first, after this parental generation, you will not be knowing the, genotype of the females of this particular group. Why? Because those females who would be having, would be carrier, they also be red-eyed. So after this parental generation, we can know the genotype of white-eyed females, we can know the genotype of red-eyed males, we can know the genotype of white-eyed males, but we will not be knowing the genotype of females. We know that these females in F1 up to F5 or in subsequent generations will either be W plus type, or they would be W plus and W type. So their exact number of carriers will not be known. And because of this reason, we can utilize the red-eyed males and white-eyed males to analyze this data. So in this slide you can see what I have done. I have taken only the red-eyed males and white-eyed males. Why? Because at least we can know the genotype of these two types of individuals. Males, they will be XY type and they will be W plus. And here white-eyed males, they will be XY type and they will be W, carrying, they will be carrying W allele. So at least we are knowing the genotype of these two types. What we observed that we took 5 males. So out of 10, their number was 50-50%. You can say that this W allele, its frequency is 50% in the parental generation. Then in F1, we observed actually we can go, means here you see 460, in F1 460 males are red-eyed and 390 are white-eyed. Then 525 are red-eyed and 210 are white-eyed. So I'm using this data, only this data present in these two columns I'm using. So when we count the total number of males of F1, then this comes 850. 850 you get after adding these two values. So if you add these two values, it is coming 850, and out of 850, 390 are white-eyed males. So it means its percentage is 45.88. Then if you count the total, add up the number of males of F2, whether red-eyed or white-eyed, this number comes 735. Out of this 735, the white-eyed males, they are 210. So this percentage is 28.57. This way you can find out the percentage of white-eyed males. White-eyed males means those individuals who carry single W allele. So here we are actually observing percent W allele in the population, in at least in the male population. Okay, only in the male population. So it is 50% in the parental generation, then 45.88 in the F1 generation, then 28.57 in F2, then 19.67 in F3. So you might have got the idea or point that only male individuals are being considered and their total is being done for every generation, and out of such total the number of white-eyed males, this percentage is being computed. So what you are observing, that the frequency or percentage of this W allele is going down in subsequent generations. That is, it is 50%, then 45%, then 28%, then 19%. It is coming nearly 15%, then nearly 12%. And if you go on continuing this experiment for further generations, then a time will come when you will not be getting the white-eyed males at all. Even white-eyed females will not be there. But for that purpose, you will have to continue this experiment for nearly 25 or more than 20 generations. See, this kind of experiment has been done by a number of workers, and they have finally observed that the white-eyed individuals completely disappear from the population. And the same thing, although this graph is not so smooth, because I have just tried to show it on X and Y axis. Here the W percentage of white eye allele is shown. Here it is 50%, then 40%, then 30%, then 20%, 10%. Here generations on this X axis, generations are there. 1, 2, 3, 4, 5 generations are there, and you will find that there is a steady decrease in the frequency of this W allele in the population. So based on this data, this kind of graph can be plotted. And but question is that why this is happening? Why one finds decrease in the number of white-eyed males or females? Actually, this can be verified by doing some other experiment. but this happens only because white-eyed males, they are not so efficient in mating process. And it has been observed that red-eyed females as well as white-eyed females both prefer to mate with the red-eyed males. So it is only because of mating preference, that is the red-eyed females and white-eyed females both prefer to mate with red-eyed males, and that is the reason. And there may be, you know, fecundity and survival reasons also, that is those individuals which are mutant, which are white-eyed, their fecundity is less, particularly their females, they show less fecundity, and their survival is also less. So because of that, there is elimination of white eye mutation from the population. So such kind of experiments, means mating experiments, have been designed, and it has been observed that the efficiency or mating success of white-eyed male individuals are very less in comparison to red-eyed males. And there is also preference from the female side for red-eyed males. So this is a kind of natural selection. So if you talk about types of selection, and then this could be stabilizing selection or normalizing selection, in which intermediate types of individuals are selected. Then you might have studied about directional selection, that is selection which favors values of a particular trait at one end of its distribution. Then the third one is disruptive selection or diversifying selection or centrifugal selection, a selection that favors extreme values of a trait at the expense of intermediate ones. So if you see such kind of thing through graph, then here in this graph you see, suppose a particular trait is being observed. I'm going to take up, suppose, the case of you know human. In case of human, birth weight, what we observe, that those individuals who have intermediate values, they survive better. So individuals having intermediate weight at the time of birth, or children, babies which are born, if they have intermediate size of um, means weight, then they have better survival than those who have very less weight, or very high weight. So that can be one case. Or if you consider suppose a rodent population, then in such population you may have individuals having very less weight or very high amount of weight, or there may be mean value of weight. So if mean value is being selected, if there are more number of individuals close to the mean value, then you can say that it is a kind of selection which can be referred as normalizing selection or stabilizing selection. So stabilizing selection means the intermediate types are being selected. Okay. Now, but if you have those individuals more selected in the population which have value either to the higher range or towards lower range, then that will be directional selection. Like in this case, see if this is the data from a population where all sorts of individuals are there and there is a mean value. Suppose this mean value is 10, but in other population, if you find that there are individuals who have a different range of variation and the mean value in that population is 15, then it means the value is shifting towards larger side. So that will be a kind of directional selection. And we can say that our increase in our brain size from this australopithecus to homo habilis to homo erectus to homo sapiens, means we have seen a steady increase in the size of our brain. So this is one of the best examples of directional selection. Okay. so where a particular trait is being selected only towards one side. Then if intermediate type is not being selected and individuals having values of lower side or higher side both are being selected, then that case will be disruptive selection. So if you have two separate populations, where suppose I'm talking about rodent again, that is those individuals who have less means weight of their body, and then those who have high weight but intermediate weight, means such individuals are very less in number. If such situation is there, then it will be a case of disruptive selection. So this way selection could be of varying types, and this disruptive selection or directional selection, all these may lead to speciation. That is, there may be two separate populations with varying traits, and in long course of time, if they get separated from each other and go on accumulating variation like that, then they may form two separate species. So such situations lead to speciation. That is why considering this aspect becomes important. Then the third factor which affects gene frequency in the population or that affects Hardy-Weinberg equilibrium could be migration or gene flow. This is very common in animals. In a number of animal population, there may be movement of individuals from one area to the other, and it's not only movement, that is the individuals, certain individuals of one population may migrate to other population and get mixed over there. They may establish reproductive relationship over there, and in such conditions definitely the mixing of two population will affect the gene pool. So the gene pool of an organism never remain intact. There may be movement of organisms or gametes between populations that may lead to gene frequency changes in the population. Multidirectional movement of organism among the neighboring populations would lead to genetic similarity among them. Now, uh, we can see how distinctly change in gene frequency occurred by using this formula, that is pi prime is equal to 1 minus m into pi plus m into pc, where this pi prime that denotes change in the allele frequency, particularly the frequency of P, change in the frequency of P. M indicates rate of migration. Pi indicates the frequency of P in the small population, and this PC indicates frequency of P in the large population. So I have made a video on this, and I will definitely forward it to you people, and I have shown how the calculation can be done. So by using this formula, you can see that how in a small population the change in the frequency of P. If we could see the frequency change of P, then we can ultimately understand how change is occurring in Q, because P and Q may be two different alleles of a single gene locus. So if we observe change in frequency of one allele, then subsequently we can know the change in the frequency of other allele. So this is a very simple formula, and I'm going to send a separate video on this aspect, so you can see the utility of this formula uh, to know that how the change in the frequency of P and Q could be seen. Then there is an aspect that is genetic drift. This genetic drift is another important factor which results into the change of allele frequency in the population, and it occurs in a small population. Genetic drift could be seen only in small populations. So this can be explained through founder effect. This can be explained through bottleneck effect. This can be explained through effective population size. I'm again saying that there is hardly a 10 minutes video on effective population size, which I will also forward to you people, and you can see that how this effective population size is calculated. So let me explain about these three aspects. Founder effect means if a population is being founded just by few individuals. Suppose there is a very large population, and in that large population there are three different kinds of individuals like capital AA, capital A small a, and small a small a. Three different kinds of individuals are there. So, and that population is very large. And suppose the frequency of this capital A is zero point 0.6 and small a is zero point 0.4. So, if few individuals from this large population move and establish a new population, okay, just few individuals, maybe just 4 or 5 individuals which comprise females and males both. So means females will have to be there if it is sexually reproducing population, because they would be able to establish and to you know, start new generation. So if a new population is being established just by few members, and if all those few members have genotype small a, then what will happen in this new population? The uh, the new population will have individuals only with the small a genotype, means only small a allele will be there. Its frequency will become 1 instead of zero point 0.4 in its parental population. It will become 1. So very drastic change could be seen, or and you can say that there gets elimination of capital A allele from this founded population. So founder effect means a population gets founded, established by just few individuals, and since those few individuals may not represent the real genetic nature of the parental population, that will definitely influence the subsequent generations or the allele frequency in the population which will come up from the founded members. Then the bottleneck effect. Bottleneck refers to the introduction of just few individuals. That is, if large number of individuals are there and because of sudden environmental change, if there is mortality to majority of the individuals and only few are left, then the new population will start with those few individuals. So this will be referred as bottleneck effect. For example, suppose there is a population of Labiorohita in a pond, means there is a big pond where hundreds and thousands of Labiorohita are there. But during summer, if you means if there is scarcity in the water, and because of fishing also, if so many individuals are taken out from that pond and only few are left, maybe just 20 or 30 individuals are left in the pond, then the next generation will be starting only from those 10 or 20 individuals which are left in the pond. So that will also influence allele frequency. Then the third one is effective population size. What does this effective population mean? Effective population means actually a population may comprise individuals of all age group, are those individuals which are reproductively mature or which are reproductively immature. So if there is a population where individuals of all age groups are there, particularly in mammalian population, suppose in primates population, you find that there are individuals who are very young, then there are reproductively mature individuals, then there are old individuals. All such individuals will make absolute population size. So entire individuals of a population, they make absolute population size. But those individuals which are reproductively mature, and are actually involved in reproduction and produce progeny for the next generation, they form the effective population size. So if one has to define what is effective population size, then the answer will be that effective population size is actually represented by those members in the population which are reproductively mature and are really contributing to the next generation. So as I said just now, I will be sending a separate video on this aspect, and that will help you to know the calculation aspect. So all these evolutionary forces do not work singly, rather a population experiences all these forces means simultaneously. Certain populations may experience the impact of one factor more severely than due to environmental severely than other due to environmental effect. And all these evolutionary factors do influence change in the gene frequency. But see, there are certain cases which I must explain. Genetic drift, about which I was just talking, is significant in a small population. I just told it. Genetic drift causes allele frequency to change randomly. So there is no, it is, you can say that there is no particular direction, rather there is totally a non-random pattern of change in the gene frequency because of drift. Then genetic drift can lead to loss of genetic variation within population, and this genetic drift can cause harmful allele to become fixed. So let me take up some examples. Dobzhansky performed certain experiment by using Drosophila pseudoobscura, and I'm going to explain that specific experiment to see the frequency of ST. ST stands for standard gene arrangement, and this PP stands for an inverted gene segment uh, in inverted area. PP means Pike's peak is the name of inversion. In a small and large population means I'm going directly to this experiment. Okay, you see this is a graph which will explain the entire story what Dobzhansky did. Actually, if you observe the polytene chromosomes in Drosophila pseudoobscura, particularly the third chromosome, then there is an inversion which is referred as PP, that is Pike's peak. So the frequency of this inversion in the population was actually observed by Dobzhansky. Drosophila pseudoobscura that carries this inversion, that is Pike's peak, which is represented by PP. He actually established several populations. See here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Means 10 populations he established by just considering, you can say, 10 individuals, means 10 male and 10 female. He randomly selected and cultured them in food bottles, and then uh, observed the frequency of P, that is PP. In he started experiment in 1955 June, and then October 1955, means July, August, September, October, after 4 months. So he could see that variations were there. He started with 50%, PP frequency, but then found variation in the frequency of this inversion. And then again in 1956, he observed the frequency of this PP in these established populations. But in every generation, he transferred just 10 flies in the food bottles. And he observed that the frequency of PP that ranged from almost 50% to, you can say, up to um, means less than 20%. Means these many variations are there. And he also started populations with large number of individuals. Again, in 10 different bottles, he put 100 flies in each bottle, males and females in equal number, and could see that very little changes were there exactly in the same time period, means in 1955 October. See here, lesser changes could be seen than in small populations, and as I told that it is around 50 to less than 20% variation in small population in November 56, but here such wide variation cannot be seen. So it is only because and if he would have continued this experiment for some more generations, then maybe in some of the population the pp frequency might have disappeared, and only st, that is standard gene arrangement or normal gene arrangement, could have been seen. So here you see lesser means narrow range of variation could be seen in these population, in these 10 populations. Although in this case, in small populations, no fixation of this PP or ST gene arrangement could be seen, but yes, obvious change is being observed. So this is percentage of PP in Drosophila pseudoobscura in small and large populations. And exactly the same thing is shown in this diagram also. That is, if just 20 individuals are taken to establish certain populations, then there could be increase or decrease, sudden increase or decrease in the in certain allele types. And if 200 individuals are taken for the same purpose, their frequency in the initial stage is 50%. Then you can see variations in their genetic composition, but there is no elimination or fixation of alleles. And if the individuals are 2000 in number, and if 2000 individuals are being taken, then very least changes are occurring in the further generations with respect to some gene arrangement. So one can do this kind of experiment by putting large number of individuals in means by considering large number of individuals in a population or a small number of individuals in a population, and such kind of drastic change in the gene frequency could be recorded. Now here another example I have taken, and this is quite interesting one. See, there is a small population in America which is known as Dunkers. This population is German Baptist Brethren. It is actually a population which actually migrated from Germany and that got settled in USA, and there is an area south central Pennsylvania, and this certain people, just certain people, few people from Germany, they migrated to United States of America in 18th century, early in 18th century, that is in, you can say, in the beginning of 1700, and then they migrated to this place, that is central Pennsylvania, and hardly um, and presently means, well, if very few people reside over there in United States of America, and they are called as Dunkers. So one experiment was done. It was not experiment, rather observation. What was done? The blood group of those people who reside near the Dunkers population, and these dunkers, they have one characteristics. They actually do not marry with the nearby people. They maintain their integrity. They try, it means marry among themselves, and they do not get mixed up with the neighboring populations. So they want to maintain their um, maintain their genetic integrity, and that is why they do not marry in their surrounding areas. So in one observation, people from the surrounding area which do not have connections with the dunkers, their blood group frequency was actually taken. A, B and AB frequency, Mn blood group, and N type blood group frequency was considered, Then Dunkers were also analyzed exactly for the same purpose, and the German population from which they actually migrated, and they were also considered for the same analysis. What was observed that in USA the frequency of A, that is blood group A, that is 40%. In German it is 45%, but these people they have this frequency 60%. B or AB blood group, its frequency is 15% in United States of America, particularly people who reside in the peripheral part area where Dunkers live, and exactly 15% is there in German, 30% in USA, 30% in German, but 45% here. Here there is only 5% frequency of B or AB. Yemen, its frequency is 50 in USA, also 50 in Germany people, but it is 41% in case of Dunkers. So 20 USA, 20 German, but 14 here. So we find much change in the frequency of A, B, or Yemen blood group types in Dunkers than the population from where they actually migrated, and also the population which live in their surrounding. So this shows that this variation is only because of the genetic drift, means these people came in just few number, and since they did not get mixed even till today, they did not get mixed with the surrounding population. They have a different genetic makeup than their parental population or the population close to which they are residing now. So these are some of the things which I considered I must convey to you people, so that you can have the idea that what are the factors which influence the you know uh, gene frequency, particularly Hardy-Weinberg equilibrium, and you can say that these are the four major forces of evolution, that is mutation, selection, migration, and random genetic drift. And for effective population size, and how to calculate the effect of migration on um, change in gene frequency, particularly in a small population. For that separate material I would be sending. Okay.